Hello everyone, this is Darshita Singh, and today we are going to cover life processes, the further thing which is left. So, till here we have covered experiment 10.4, we have covered and some fascinating facts. Yeah, and we have done recap. So, now we are going to cover the next part, that is experiment 10.5. This experiment is all about identifying the gas which is released during photosynthesis. So, we all know that. Photosynthesis is a process of synthesization of food wherein plants make their own food. So, this equation is very important when it comes to photosynthesis. This equation is carbon dioxide plus water in presence of sunlight and chlorophyll gives oxygen and glucose. So, this is what the whole photosynthesis process is, and it is summarized in this one equation. Now, let's see that this oxygen is actually released or not. So, this is the experiment. You have to take a potted, so you have to take a conical flask, sorry, a test tube, and invert the test tube and place a particular water plant in it. Then this experiment is made and this whole sorry experiment made this whole setup is made and identical setup B is also made. So now what you have to do, you have to keep setup A in sunlight and setup B in dark. The setup A is observed, so setup A has all the above things like carbon dioxide, water, sunlight, air, chlorophyll, etc. So it will start the process of photosynthesis. But this is the setup B does not have sunlight, so it will not perform the process of photosynthesis. So in this setup A, you will start seeing bubbles which are emerging from this setup. Okay, and when you lit a mastic क्यों lit कर रहे हैं हम लोग because it is an indicator of oxygen. So if you lit a mastic here, then it will produce an intense flame. Whereas in setup B, there is no gas bubbles or oxygen present, and there will be no flame if we lit a mastic here on the start of this test tube. So what we have concluded here is that oxygen is released during photosynthesis, and this whole process occurs in the presence of sunlight. Okay. Now let's move to the next experiment, experiment 10.6. This experiment is about the gaseous exchange of plants. So you have to basically, this experiment can't be shown with images. You have to conduct this experiment in your biology lab only. But let's do if it's given in your book. So you have to take, you have to collect a leaf, then peel the thin layer from the lower surface of the leaf. Then you have to drop some water, put that peeled leaf on a slide, cover those leaf with color cover slip, and under the microscope you will observe the small tiny pores called stromata. These are stromata, and the other two guarded cells are guard cells. This is guard cells. Yes. Guard cells, what they do, they protect stromata and helps in opening and closing. Opening and closing of stromata and also protects stromata. Okay? Okay. Now let's recap of what we have done. We have only covered two experiments here, experiment 10.5 and experiment 10.6. Now in this experiment The conclusion is that oxygen is released during the process of photosynthesis. And in experiment 10.6, we learned about stromata, a tiny pores present on leaves which helps in gaseous exchange. Okay. Okay. Now activity 10.7. This activity is demonstrate that plant has this upward movement and water moves to from roots to other parts of the plant. Okay, so this can be shown by this experiment. You have to take two tumbler. A tumbler is with water and B tumbler is water, and you have to put some red ink into that tumbler. Okay. Now what you'll observe that after like few hours or after like next day 24 hours, you'll observe that the red color will be visible to all the parts of the plant. Okay, it will be observed in leaves, flowers, stem of the plant. Okay, so now what does this demonstrate? This demonstrates that the ink that was like put in the tumbler B, it moved upwards, and this upward movement is due to a small tissue called xylem. Okay, this is xylem. This particular tissue is xylem. So this tissue is a thin network or tube-like structures in the stem only and in branches and leaves also. So this actually helps in water and mineral transportation. Okay. So xylem tissue is basically responsible for water and mineral transportation because these are the things which roots are responsible for taking care of, right? Root take care of these two things. Roots say they go up and to other parts of the plant, especially leaves, because leaves they have to prepare food. So yeah. Now we'll see कि food बन गया है, how the food will be transported to the rest of the body of plant. So food is prepared. Now leaves have to distribute it throughout the plant. So there is another tissue called phloem. Phloem transports the food from leaves toward the plant, and this is also a tube-like structure. Okay, and plants store food, which is in form of starch, are present in seeds and roots of plant. Okay, so again, what we have learned about activity 10.7 in which there is this experiment of showing xylem tissue. Okay, and then we have learned another tissue called phloem. This tissue is present in stem of the plant, which carries those food which is prepared by leaves to other part of the plant. So food to other parts of plant. Perfect. Now the last activity, activity 10.8. This activity demonstrates the release of carbon dioxide during respiration. So now we are done with food production and all that. Now we are going to see that plants do they respire or not. This experiment is all about the plant releases carbon dioxide during respiration or not. Okay, so the respiration and food production, that is photosynthesis, these both are different activities. So in respiration, they release carbon dioxide, but in photosynthesis, they release oxygen. So अगर difference करने को आएगा, so please write that. Okay, so साथ में yes. So we are going to learn activity 10.8, which signifies carbon dioxide release during respiration. So this in this particular experiment, you have to soak some mung beans in overnight, and then you have to put those into a conical flask. Okay, this is the image of a particular activity. Then this whole test tube connect to test tube A and test tube B. These are the germinating seeds. Okay. Now the test tube B is connected with another test tube. Let's name it C. And this test tube C has lime water in it. Okay. I'll tell you why lime water is put here. But this test tube C is also, it also has lime water, and B test tube is also, it also has lime water. Now what you will do, you leave the whole setup for 24 hours in dark, and now what you will see is you will compare the lime water in the test tube. Okay, so the lime water in the test tube, which is connected to the flask, that is test tube C, it will turn milky. Why? Because this indicates that carbon dioxide has increased. So what is the indicator of oxygen? Oxygen का indicator was कि inflammable flame was created when matchstick was lit. But for carbon dioxide, the lime water turning milky is the indicator. Okay? Yeah. So you'll observe that test tube C will turn milky, and you'll compare those two lime water, in the lime water which is present in D and the lime water which is present in C. You'll compare those two lime waters, and you'll observe that C wala lime water is turned milky. So this is how we have concluded that carbon dioxide is released during respiration of plants. Okay, so what we have learned till now, we have learned about activity 10.8 only. In this, we have learned that plants respire and they they exhale carbon dioxide. Also, this is the equation of respiration. Glucose plus oxygen gives carbon dioxide, water, and energy. Okay? And this energy is released during respiration and helps in plants development. Okay, now these are some questions, and this is the Google Form link. I'll put this Google Form link in the description as well. You can actually do the test and check your progress till now, and that's it. Please do these questions. These are some good questions. Please do these questions in your notebook. And that's it. Thank you. Have a nice day. The chapter is over. Thank you. Bye bye.